So what are we going to do today? What I'd like to do is just kind of give you some background on just bioinformatics in general. I think we all have an idea what bioinformatics is, but I think if you really sat down, you probably don't really know what it is. So what we're going to do is kind of go over today, some of the stuff what are the data we use for bioinformatics? What are some of the interpretations we can get out of that? Then we're going to have an interactive demonstration. Did everybody bring their computers today? Good. Yes. Okay. It's bioinformatics, so bioinformatics requires computers. So thank you. If you didn't, you can just watch. And like I said, this is going to be recorded, and hopefully, if you missed something today, I'm not requiring you to take notes or I don't pass out handouts. What we'll do today is I want you to kind of be in the present moment as you kind of go through this, right? I don't want you looking at other things. I just want you to kind of have fun today. And if you want, you can go back, because of this recording, you can go back and actually, if you miss something, you can watch the recording again and do it again. So we'll have an interactive demonstration. We'll all kind of do the same thing. And then if we have time, we'll do some independent exploration. So I'm sure you all have cancer projects on your own right now. So basically what I'm going to show you today are tools that you can use for your own research. So as we go along today, what I want you to do is bookmark all these pages or these websites that we end up on, okay? And then at the very end, maybe we can discuss our results. So my first question is, what is bioinformatics? And I always ask this of my classes, it's interesting the kind of responses I get. So what would you feel you would use bioinformatics for your research? I'll start with you.
So with cancer, looking at genetic mutations and classifying them and grouping them with patient outcomes.
Okay. Yep. Absolutely. What about you?
Presenting tables in like a more interesting way.
That is definitely part of it. I would say a lot of biostatistics kind of does the same thing, but yeah, that's definitely part of it. Going down.
Could you use it for like sequencing genomes?
Absolutely, that's exactly what we do. So a lot of what I do in bioinformatics is interpreting sequencing data, right? And that has its own problems that we'll talk about.
Creating libraries of different. We have a CRISPR library thing, and you can create those, sequence those, decide what's best in that library that works for your experiment to put on your cells, I guess, bacteria, whatever you're working with.
Okay. So it helps you genetically modify certain things, right? A lot of blast searches, a lot of, does this sequence compatible with this one, right? Totally.
Which mutations correspond with different grades and stages of disease?
Absolutely. That is a huge part of bioinformatics, is taking the actual raw data and connecting it with the clinical data. Absolutely. You.
Yeah, I would just say, I guess, for my project at least, maybe just like data organization.
Okay.
Or like making graphs.
I will say most of what I do, probably 70% of all my effort is spent formatting the data just so I can look at it. So it's not, it's not all cocktails and, parties, right? Most of it's me sitting in front of a computer, usually with a beer, at night, sitting there reformatting the data so I can get it into a spot that I can actually look at it. Well, here's my simplified version of bioinformatics. And this all came to me when my children were small. Actually, my oldest just completed her first year of college, but we used to go to Denny's or IHOP, and they used to give you those placemats, and they'd have little games for the kids, right? And usually one of the games was they'd have two pictures that looked very much alike, and they'd say, all right, please, circle what's different in them, right? And that's kind of how I see bioinformatics, is that if you look at actually the sequence of most human beings, we're only 99.4%, we're 99.4% identical as far as the sequence goes. So for the most part, we all look, from a genetic standpoint, we look pretty much the same, but there's obviously differences in that 0.6% that can cause diseases, can cause huge phenotype changes, make people look completely different, right? So in actuality, what I am doing, or what I feel I do, is I look at two things, say a disease group and a group that's normal. And what you're trying to do is find a pattern that's in one that's not in the other. At its deepest, like the most purest definition of bioinformatics, it's pattern recognition, right? That's all it is. And that's why I do love teaching bioinformatics to kids. Like, I've taught high school kids this. So you can absolutely and utterly do this. Because young people get this. You guys play video games. Like, pattern recognition is deep down. You grew up with computers. So for you, this is a lot easier than, say, me trying to teach a 60-year-old professor how to do this stuff. It's terrible. They usually think this is voodoo. No, it can't be. That's too easy. Like, it's that easy, I'm telling you. You know, you can kind of think of all this data as as pixels, right? You know, if we look at the human genome, I mean, we're looking at almost 6 billion bits of data, right? I mean, you can kind of think of these things as pixels, right? How many pixels are in your average high definition TV? Does anybody know? Any guesses?
1080?
No. Actually, in your 4K TVs, it's about 8 million pixels, right? So when you're watching your TV, do you see those boxes? No, of course not. If you do, you should seek help, because that's not normal. Or work for the government or something. I don't know. What I see is, you know, these pixels. What our mind does is actually puts that in together to form a picture, right? And as you're watching your TV, you know, basically you're watching boxes, and they make you laugh, they make you cry, you know, they make you do all kinds of things. But in reality, if you want to get to the truth of it, you're basically watching zeros and ones, right? So as a bioinformatician, it is my job, or how I feel it's my job, to basically take all these pixels and put it together in a comprehensive picture that I can say, go to my collaborator and say, hey, listen, you know, this is your disease, this is what I think is going on, this is what I think you should target. Does everybody understand that? Okay. So where do all these pixels come from? And these pixels are getting generated exponentially. Like, every day, it's just a waterfall of information coming. And if you look back, honestly, you know, all the scientists in the world could stop, you know, sacrificing mice, running Westerns, RT-PCR, and just with the data that's already on hand, I'd have enough for 3 careers easily, right? There is tons of data. And what you want to do as a bioinformatician, it's my job, or what I feel is it's important for me to actually utilize that data, right? What cancers are you guys working on? Leukemia?
Triple-negative breast cancer.
We're going to go over that today. It's going to be sweet. Yeah.
Rhabdomyosarcoma.
Yep. Melanoma? Breast? We're going to go over breast today too, which is triple negative. Yes. You know, you're not the first person to actually investigate this disease, right? And how many samples can you actually generate, right? You know, maybe you get a grant, you know, maybe even if it's $100,000, I mean, you know, you've got a limited clinical thing that you can actually look at. There is data out there so that, you know, if you're working on breast cancer, you can access all these other people that have actually done studies on it, right? And that will make you better than your lab mate who is just looking at only the data that they can generate. Bioinformatics is an absolutely and utterly crucial tool to help you with your research. It's basically like getting the cliff notes before you actually do your experiments, right? I go in there, I'm interested about breast cancer, I go find a study that kind of looks, say, triple negative, I start looking at that, I generate hypothesis, then I can develop a very specific experiment or study to address that. Does everybody understand that? This is huge, and a lot of people don't understand this. So you're way ahead of the game right now. So where does all this information come from? So when I was in grad school, we used to bow to this. This is the central dogma of biology, right? Transcription, translation, protein, right? And we could take a look at this. Just with this information alone, it's a ton of information, right? We've got DNA, again, I mentioned you've got 6 billion bits of information that we can analyze. We can sequence all of this. In humans, there are approximately 25,000 genes. Again, we can basically isolate that messenger RNA and count or basically quantify the message. And then from the RNA, you have translation and you get proteins. And in its purest native form, you probably have about 500,000 proteins in humans, right? Pretty much all this stuff we can measure. And as you can see, as you can imagine, like your average 60-year-old scientist that never did any, programming in his life was like, what am I going to do with all this, right? So that's why when I first started, there wasn't no bioinformatics. This discipline came about because we were generating so much information that even the biologists couldn't specialize in that, right? You needed somebody that actually specialized in looking at all this information. But this isn't it, right? We also have microRNAs. Everybody's heard of microRNAs, right? Okay, good. Usually when I talk to high school students, they're just like, micro what? Is that a robot? No. Basically what these are little bits of RNA that bind to the messenger RNA and that basically regulate it, right? It can cause its destruction. So this is another level of regulation that we can also measure that impacts what this organism is going to function, how it's going to function, right? Even with the mRNA, right? So just because there's 25,000 genes doesn't mean there's 25,000 different versions of mRNA, right? mRNA has introns, exons, right? You can shuffle them around, you can change them. So I was working on one gene that had I worked on one gene that had 11 versions of its messenger RNA, and each version did something different. It was an absolute and utter nightmare. I hated it. You're like, oh, what's up? Oh, which version? Oh, crap, we weren't thinking about that one. Right? So you have complexity even in the messenger RNA itself, right? You can have different versions that do different things. All right? Proteins are a mess. I don't know. Does anybody work on proteins? Anybody specialize in proteomics? I did a postdoc. I got the heck out. There's just so much stuff you can do. You can ubiquitate it. You can phosphorylate it, right? It all changes the function of these proteins. So it's very hard to keep track of what's going on. But again, we can measure all of these things. Let's go back to the DNA itself, right? DNA has structure, and I don't think a lot of people really realize that the biggest factor in whether a gene gets expressed or not is not a transcription factor. It's not any of these signaling molecules. It's the accessibility of that DNA. It's structure, right? If it opens up, then that machinery can get in and copy that DNA. Right? So it is structure. So structure plays a huge role, and we can measure that, right? You can methylate certain areas of the genome and basically have a completely different phenotype. And a good example of this is all our cell types, right? Every cell in your body has the same DNA. Is every cell type the same? No, right? Certain genes get expressed on and off. Me doing this, looking at gene expression, I would say probably in an average human, average tissue, probably only maybe about 40 to 50% of the genes are being expressed. That's it, right? So a lot of this compacting, the epigenetics is what it's called, right? The structure of the DNA plays a role in understanding the biological system. And again, we can measure these structural changes. So what do we do with this? And in its simplest form, what we're really doing, you're just taking, I have a group of people that are normal, and then I find a similarly matched group with a particular disease. What I'll do is I'll apply some mathematics. This can be very simple, or people can make it very, very complicated, trust me. I usually find simple works better, actually. So you can do all kinds of tests, parametric, non-parametric, but what you're eventually doing is what you're generating is a gene list, right? You're getting a list of genes that are different between two conditions. For me, this is where the magic starts. This is the art of bioinformatics, is because a lot of times when you look at, say 2 conditions, you'll find thousands of genes that are different. How do you make sense of that, right? Do I just take the top gene, the one with the greatest change? Do I take groups of genes? What do I do with that information? So today we're going to actually go over basically what I feel is the key point of bioinformatics is the interpretation, right? I got two systems. I found out what's different. I've got a list of things that are different. What does this mean? Okay, does everybody understand that? And it's not only just finding what's different, but it's also finding what's similar can play a huge role in interpreting your system. So you'll notice on this side, there's two genes here, right? One on top, one on the bottom. The height of the bar, so each one of the bars is a particular sample that they measured the expression of this gene. The height of the bar relates to the amount of expression. So the higher the bar, the more it's being expressed. The lower the bar, the less it's being expressed, right? All these samples correlate to each other, right? So if you look, you have two genes here. Here, it seems to be really high, and then it seems to go down. You see this kind of low, then it goes high, low, and then you could go over here. You'll see high or low, high, low, really high, low, low, high, low, really high, low, right? Seems to have kind of a similar pattern, right? What do you think of that? What does that mean with these two genes?
Expressed across different conditions.
Yes, they are. But what would that maybe indicate? What do you think is going on there? Why are they being expressed at the same time?
Correlated.
They're correlated, but do you think, basically what I'm trying to get you to see is they're probably performing a similar function, right? That it's like they're super friends, right? They're best buddies. That if you saw two people, like you knew two people, and whenever you saw one, you always saw the other. But if you didn't see one, you never see the other. Would you think they're friends? Probably, yeah. And that's what kind of happens with genes. Genes that behave similarly are probably involved in similar functions, right? And we can utilize this information. Say, I don't know what a gene is, but I find out what all its buddies do. I can kind of figure out what that gene is doing as well. Do you understand that? All right, well then you can have the opposite of that, right? So look over here, you've got really high gene expression here, and when it's high here in this gene, it's low, gets high here, gets low, and so on and so forth, right? What do you think's going on there? An opposite pattern.
Opposite functions.
Right? Maybe one is suppressing the other, right? And that happens a lot, right? Maybe they're the Legion of Doom, they're enemies, right? You've got these genes, this gene can't get expressed until this one goes down, right? Maybe it's suppressing its transcription factor that's causing its transcription, right? And vice versa. When this goes down, this one goes up. I see that a lot, and this is a very critical pattern that most people don't recognize in the data. This is another aspect of the data that you can actually utilize to understand your system. Okay. So over the years, I found the easiest way to understand bioinformatics and kind of the search that we do is to kind of frame this as an investigation, right? So what I'm going to do is I'm going to kind of go through kind of an example of a bank robbery and how basically use these tools to figure out who might have committed the crime, okay? And I'm also, as I do that, I'm going to give you examples out of my own research. So I used to work in the lung spore. I don't think it's here anymore, but basically I worked a lot on lung cancer. So a lot of the examples I'm going to give you are going to be based on lung cancer. So, all right. So we're an investigator. All right. So in the middle here, there's a bank, right? The bank and all of these things, these are genes or proteins. But in actuality, you can think of them as people, right? And every one of these people, if you ask them who are saying, hey, I was by myself, I was walking by the bank, minding my own business, it was robbed, I had nothing to do with it, right? Everybody's going to say that, right? So we have the bank. It gets robbed, and as soon as it gets robbed, shortly after that, they basically shut down the whole area. So basically, everybody's kind of frozen to where they were shortly after the bank was robbed. Where do you think most of the robbers are going to be? What's that?
I said the furthest away.
Well, they're going to try, right? But remember, it happened like right after it was robbed, so everybody rushes in. So where do you think most of them are probably going to be?
By the bank.
By the bank, yes, thank you. Oh, and you're the investigator. So I used to use Columbo, and no one knew that reference at all. Does everybody understand? Everybody knows who this person is, right? It's Law and Order? Okay. No?
Older.
Oh my gosh. Thank you for being here. Trust me, there's going to be lots of references you don't get in this talk. So the bank's robbed. Yes, most of the robbers are probably, if they lock it down really quickly, most of the robbers are going to be by the bank. And these are represented, the ones in the red, right? Some of them, maybe one was a track star and ran really fast. Maybe one of them had like a scooter that was nearby the bank and got away a little farther. But the closer you get to the bank, you're probably going to encounter more guilty individuals, right? And we utilize this in statistics a lot, right? What we do is we can draw parameters around that bank, right? So if I have a p-value, does everybody understand p-values? Everybody's used p-values. Basically, everybody knows p-value 0.05 less than good, I think, right? Yeah, of course. I can have a whole talk on p-values, but for this discussion, let's just, okay, so we're talking, we draw the perimeter, we're basically taking all the genes that are less than 0.01, right, a probability, right? So we do this and we draw this perimeter, right? But as you can see, probably the majority of individuals are guilty, but you can see there is some, innocent bystanders in that group as well. And that happens in our gene list. When you take a p-value of 0.05 or less, just remember there's a lot of error in that. And the fact that we've done, I said humans have about 25,000 genes. We're doing 25,000 statistical tests. So we will generate error. So there will be false positives, right? We can draw this parameter anywhere you want. You don't have to use a 0.05, right? That's just something people just made up. It's an arbitrary value. I could go out to 0.1, right? As I extend that perimeter, I'm getting more guilty individuals, right? Exactly. And that's the trade-off that you have, right? And what most people do, has anybody heard of false discovery rates, FDR? No? Oh, man. Yeah, not that FDR. Not the president. False discovery rates. So what most people do, so a p-value is just specific to a particular gene. False discovery rates have to do with gene lists. And what I say is if I have a false discovery rate of 10%, what I'm saying is that I've got this gene list and I only expect 10% of those values to be false positives. Does everybody understand that? So what most people do is they correct for that. Since we're running so many statistical tests, what they like to do is draw their circle really close to the bank and say, all right, through my statistics, I figured out that, if we draw it here, only 10% or 1 out of 10 is going to be a false positive. And that's great if we're looking for say a biomarker, right? If you want something that, you want to use as a marker to determine disease, say severity or something like that, you want to make sure that what you have is true. But a lot of times, trying to understand the system itself, right? I've only got all these guilty individuals. Maybe that's not enough to understand the system, right? So it all depends on where you draw the perimeter. It depends on, what you're trying to get out of your data. Does everybody understand that? And it could be a huge thing. I think as you go into a project, you have to keep in mind, what am I really searching for? Am I trying to understand my system, or am I trying to find something, say, as a biomarker or something that I know is there, right? Okay. Oh, did I? Okay. Oh, I took it up. Oh, no, I didn't. Okay. All right. So again, we already decided we have our investigation, bank's robbed. We know that there's going to be a higher density of guilty individuals closer to the bank. And you're the investigator. You have access to databases, Scotland Yard, why not? Anything you want, right? CSI, whatever you want. So not only just location, what else would you use to determine the guilty individuals or not? What's that?
Security.
I never thought about it. You're the first one to say that. There was no security footage. They disabled the cameras before they robbed the bank. But what else? I mean, you've got all these individuals here. You can ask them anything you want. What else? What else would help you determine if they're guilty or not?
Consistency and coherence of their story.
Could be, yeah. What about criminal history? Right? I know we all, hey, I'm a hippie too, but if somebody's been convicted of robbing a bank 5 times before this and they say, hey, I had nothing to do with it, you're like, yeah, probably not, right? And that's what you can do. You can actually look at the criminal histories of these individuals. And some criminals with a criminal history might not be associated with this crime. Some individuals with no criminal history might be associated with this crime. But what it does is it kind of allows you to basically kind of figure out who are the usual suspects. Has anybody seen that movie? You have. Okay, good. Usually I show that and I'm like, I haven't seen that. I think Kevin Spacey messed it up for everybody. No one wants to see it anymore. I know, he's a weirdo. Okay, so, you're looking for the usual suspects in this, right? It's like, and there's not just one bank, right? There's just not one form of cancer. There's lots of forms of cancer, right? And say if I'm looking at my gene of interest, I think it's involved in my cancer, and it's also been around like 2 other banks that have been robbed at the same, at other times, that's probably a good suspect, right? And that's what we do in bioinformatics all the time, right? Is basically what I'm looking for is I'll get a gene list, I'll start going through it and looking to see who is, if I'm looking at cancer, what genes have been associated with cancer previously. And that kind of gets us to what are the usual suspects in cancer, right? And I think everybody is focused on oncogenes, right? The criminals, right? What are some oncogenes? Anybody give me some oncogenes? KRAS, yep. MEK, yep.
BRCA2.
That would be more of a tumor suppressor.
Oh.
Yes, totally. And we're going to talk about that today too. Yes, what are some of the oncogenes? Growth factors, EGFR is typically, I've seen progesterone be a factor in cancer. Signaling molecules, a lot of things got to go on during cancer. Cell cycle genes, right? Critical points of, things that stimulate the cell cycle or inhibit it, or basically stimulate it, right? A lot of things like that. Angiogenesis. If you're making a tumor, if you have a tumor, it needs a blood supply or it's not going to grow. So you have genes that supply it blood vessels. Things that maintain telomeres. I see topoisomerase is upregulated in a lot of tumors that I look at. Anybody heard of topoisomerase? You know what that is? It's maintaining the ends of the chromosomes. Every time you divide your chromosomes, you lose a little bit at the end of the chromosomes, right? And if a cancer, that's horrible, because then you're going to eat into the genes and then the cell's going to die. So what these cancers do, it upregulates the creation of the telomeres so that you don't lose any bits of vitality in the tumors. And again, some of these are KRAS, EGFR, MYC, VEGF, which is the angiogenesis component. But what I don't think is a lot of people don't focus on the tumor suppressors, right? These are the cops trying to keep the bad guys, at bay. By the way, these oncogenes, do they start off bad? No, they're genes, right? It's like something happened in their environment that corrupted them, and then they turned to the dark side, right? Like if you smoke, you're basically creating a criminal environment in your body that will turn some of your genes bad probably, right? But you have mechanisms, and the reason, some of these oncogenes, I'll tell you right now, probably everybody in this room's had cancer. The only reason that we're all still here is our body, our immune system takes care of that, right? Our tumor suppressors take care of it before it becomes a problem. So what are some examples of tumor suppressors?
P53.
P53 is the biggest cop for sure. What else?
Said that a little before.
You did BRCA1, yeah. What else? Name another one. Apoptotic. Did anybody know any apoptotic genes? P10 maybe? I don't know. When you look at genes like all day, like they become your friends after a while. I've had discussions with genes. I have my favorites, and yeah. I need to get a life. Okay. So you've got like cell cycle inhibitors, right? You have things that are trying to shut down the cell cycle, DNA repair enzymes. We were talking BRCA1, telomere maintenance, things that are trying to stop the telomeres from getting overproduced, apoptosis, programmed cell death. And again, some of these are P53, BRCA1, which is very associated very much with breast cancer, retinoblastoma 1, and P10. Right? But what I want you to understand, and why I'm showing you all this, is that it's not just oncogenes and tumor suppressors. I've seen so many labs try to basically narrow down a cancer by defining it with one gene, like a oncogene or a tumor suppressor, right? This is about P53 or this is about KRAS. It's never that. Basically, cancer is basically the fight between these two different forces, right? And that's what you need to understand, is that the oncogenes are fighting the tumor suppressors, are trying to blind them, and the tumor suppressors are fighting the oncogenes. But it's this tug of war here that we need to pay attention to. And it's not just not the criminals and the cops, it's the interactions of both which are going on in your system. That's what's important. All right. So I'm going to talk about one of the criminals I found in my research. Its name was polo-like kinase 1. Has anybody heard of that gene before? I didn't either. I had no idea about it. But I had a collaborator come up to me. He was working on lung cancer. He's like, hey, we know this polo-like kinase 1 is very important in our system. We see it like upregulated in all these massive bad cancers. We don't know what it does. Like, give us some insight into what's going on. So, you can just kind of look up this gene. Obviously, so what, first thing I do is let's look up its criminal history. Is there a history here that might say that this might be a huge player in lung cancer? So we look, cell cycle, yeah, that's a hot topic. Trigger for G2M transition, absolutely. Helps separate the chromosomes, and then it basically, if you lose it, you induce apoptotic pathways. So it's preventing cell death. And tumorigenesis, look at, has it committed other crimes in other cities? Oh yeah, it's committed lots of crimes. Like it's big time and elevated in many types of cancers. And this is what I'll show you right here. This is a program I use. It's called Cohort Analyzer. I wish I could let you guys use it, but all the really good stuff is not free. Yeah. And even I get around it too. What I do is I end up doing videos for them. That's why I have a YouTube channel. So I'm like, hey, I'll do a video for your thing. Can I have a free version? And they go, yep. So I have a lot of software for free, which is cool. So here, basically what this is showing you is basically these are different cohorts containing these are the diseases on the left, and over here is 100% of the samples. If it's red, that means in those percentage of samples, this gene was overexpressed, and if it's green, it was underexpressed compared to normal. As you can see here, if we go through all of these things, we're seeing a ton, like in most of the samples, especially like bladder cancer, look at this one. Almost 100% of the samples have elevated levels of polo-like kinase 1, right? And if we actually go down to the lung cancer one, do a close-up here, it's like 97% of these samples have some alteration of polo-like kinase 1. Either it's upregulated or you have you actually have multiple copies, right? Basically what I'm saying is we got a good criminal. Based on the criminal history here, I think we definitely got somebody involved in the crime, right, of lung cancer. So what kind of crimes is this person or this gene committing? Like the worst one, which is murder, right? If we look here, this is everybody's familiar with Kaplan-Meier curves? Maybe? No? People use them all the time, and they get overused, and normally, sometimes people interpret things out of them that aren't necessarily correct, but sometimes Kaplan-Meier curves can be good. But anyway, so it's basically a survival curve. And what this is showing is what we do is we break up the expression. So we'll take, basically divide the the samples in half. These will be the high expressing cancers, and this will be low expressing cancers of pill-like kinase 1. So here you'll notice when it's low in the purple, the survival over 5 years or even longer is much better when you have high polo-like kinase expression. So something about this gene, and it's being overexpressed in these samples, is contributing to the actual death of the patients, right? Good criminal. Well, bad criminal, but good in our case. And we can go way back into history. We can basically say, these cancers that we look at, especially lung cancer, you can actually take, basically it's like a camera they put down your throat, and you basically go in your lungs and take a look. So you can basically see these lesions. At stage 7 is the only point where we're actually treating the cancer, but you start out normal, you can get basically the cell walls thicken and tighten, and you have morphology changes, and then eventually you have cancer. But if you look here, this is us going way back. We're talking to the psychologists that looked at polo-like kinase as a kid, and even back then, the cancers that got worse or persisted had a much higher level of polo-like kinase compared to those that actually regressed, right? Okay, so second example in our bank example. So we said, okay, most criminals are going to be by the bank. If we do the criminal history, we can look for people with a criminal history, and they might actually be tend to be more guilty. What other things would you actually look at, in this group here to determine who's guilty or not? Any ideas? So you got criminal history. Fingerprints. Yeah, that would work. How about this? What if there's a gang in here? Like, say there was 5 individuals associated with a particular, or there were 7 individuals associated with a particular gang, and you found 5 of them just happened to be walking by the bank as it was robbed. Do you think they might be more guilty? Yeah, heck yeah, right? You have this gang, this criminal gang. Everybody was saying they were just walking by the bank by themselves, and 5 out of 7 just happened to be by the bank when it was robbed. That's not normal, right? This is telling me that, and these are these kind of pathways we look at, right? And these pathways can be huge. You can have lots of members, be small and powerful, and it can be doing all kinds of destructive things in your cell, right? And what you need to understand is that these gang members share members, right? It's not just one gene can be in one gang. If you look at P53, it's in like 1,000 gangs, right? Because it's involved in all kinds of stuff. But the thing is once you find the gang, you can actually probably find some other guilty individuals. So if I find this gang and I know this one's individual and I know his gang's around, I'm pretty sure his gang was probably involved in it. Everybody understand that? Yeah, we can totally do that. Does anybody know who this is? Yeah. I will give $5 if anybody can name this gang. None? It's Cool in the Gang. It's a music group, which no one knows. Okay, well, that was our one gambling for the class. All right. So basically, pathways are things that we define. And it's kind of like the cops, right? Is that the cops say, okay, we've done our investigation. We feel that these people are involved in this gang. Do you think their assessment is completely 100% accurate? No, of course not, right? Cops don't know everything, right? These criminals, you know, like members come and go, right? They could just say, hey, we've defined this, but you know, we don't know all the members. But what we do is we try to find some core members and say, this is a pathway. All these people, all these genes are working with each other. And the way we find overrepresented pathways, and I'm going to give you this example, and after I do this, you're going to know more about bioinformatics than like 70% of the scientists out there. And it's very easy. This is how all bioinformatics works. Is say I have a pathway, right? And I feel that this pathway is associated, say, with my lung cancer study. I think this pathway is involved. And in this pathway, it has 40 members, right? 40 gang members that we know of, right? And I do my statistics, and I find, you know, I take, say, normal versus, you know, say, metastatic cancer, and I find 152 genes that I feel are associated with this disease, right? So the reason why this all works is because there's a limited number of genes to choose from. So if we remember, I said there's about 25,000 human genes, you know, that we have, right? And all these 40 members are part of that 25,000 genes. So imagine, and I get, my statistics say I have 152 genes related to whatever I'm looking at. Think of this, is that these 40 members, say, are pink balls in a sea of 25,000 green balls, okay? What are the odds if I had 152 draws from this 25,000, what are the odds that I, say, get one pink ball? What do you think that is? Pretty rare? Not really. I mean, if you wouldn't necessarily expect it, but to get one pink ball, not a big deal. What if after 152 draws, I get 3 pink balls? Now we're starting to get a little more. Now statistically, that's not supposed to happen, right? It can. I mean, there's always a random chance, but probably not. But here's usually what happens in statistics or in bioinformatics. Say I get 7 of these things, right? The odds of that, you know, P.00005, right? You know, the odds of, you know, that happening are very, very rare. But so given these results, given the fact that I got 7 out of the 40 in just my 152 draws, I would say that this pathway is playing a role in my system. And that's what we do for all kinds of stuff. And it can be pathways. It can be, say, genes associated with a organelle, right? You could say mitochondrial genes. What are the odds that I get all these mitochondrial genes in my gene list, right? Or genes that we know are associated with a particular drug, right? What are the odds that I get all these genes that are known to be affected by a particular drug, right? We can do all that stuff. So that's kind of what we do. And that's what I did with the polo-like kinase. Basically, I went in and I said, okay, here are the pathways. Remember, these are the gangs associated with histology. So these gangs get more powerful as the cancer gets worse, right? And here I can say, and then I could do another test and say, okay, I want to look at just those genes that are higher in the or pathways associated with persistent rather than the aggressive forms of cancer, right? And when I do that, the pathway comes up for polo-like kinase. Again, this is what the cops are saying polo-like kinase is involved in, right? And the log p-values, the odds of me getting that is very unlikely. What this is telling me is that not only is my criminal there, but he brought his gang with him. Everybody get that? And if we look at this, so here's a picture of that pathway. Everything in red showed up in my screen, right? And you can see this pathway, most of these things are in red. But you might say to yourself, like, okay, what does this do, Mike? Like, you know, how does this help us? If I'm interested in basically targeting polo-like kinase, and if I know the particular pathway, what I can do is focus in on certain things. This SLK is known to induce polo-like kinase. If I can't get to polo-like kinase, how about I try to basically inhibit this one? Or this molecule here, Pt.1, is known to inhibit the dissolution of the factor, or basically it inhibits polo-like kinase being associated with molecules that stimulate cell cycle. What if we eliminate this? Then we basically stop the stimulation of the cell cycle. Again, what I'm trying to do in bioinformatics is not only do I find a gene, but then I want to basically broaden out and find its environment so that way I can basically manipulate it, right? I can look for little avenues to get to it, right? This is my strategy right here. All right, we're almost done. We're just like tired. It's not even Monday. So we got specialized networks. So another example. Okay, we don't have a gang here, right? We've got all these individuals. Say this individual he's a criminal, but he works in a auto body shop, and he works with these 3 individuals, and this person is this person's sister. It might not be a gang, a criminal gang yet, but what it might be is something that we haven't seen before. And there's all kinds of pathways out there that we can basically, that we don't know about, right? Maybe this is special. This is a group put together on a special circumstance, right? Something we don't know about. And we can do that. So today we're going to look at string, and what I can do is in this software you're going to see today, is I can basically say, find me the things that are most connected based on your database to polo-like kinase. And it did that, right? So this isn't a canonical pathway. This is things based on a database of lots of different variables. And when we start looking at these things in here, which are color-coded, we find that most of these things are involved in the cell cycle, which makes sense, right? Here's its gang. These are the crimes that it's committing, right? It's basically stimulating cell production. We can use another program. This is Ingenuity Pathway Analysis. Anybody heard of IPA? Labs here have no money. So this is another commercial software. But what it'll do is it'll take your gene list and it'll basically make these networks of connections, but it'll also include things that, say, did not change in your gene list, just so that it can connect the dots. And that's what happens all the time. The very important things in your cell probably don't change at the expression level very quickly. That if you want to get somewhere very quickly, would you call a cab or build a car? Call a cab, of course. Actually, no one calls a cab anymore. You call Uber, right? Your cell is the same way. I need P53 now, right? I don't have time to build more P53. So what it does is it hangs out in the cytosol fully formed, and you basically remove an inhibitor. You translocate it to the nucleus to actually do its function. But what you don't do is you sit there and you make it from scratch. And that's what your body does. And that's what you find, like VEGF. I never see it change gene expression, but I know it's there because it's interacting with so many different things in my system. Right? So now we have another criminal. So what this allowed, we went after one. Now we've got another one, VEGF, associated with this crime. Right? Oop, polo-like kinase. Okay, last example for the crime. Okay, say we have all these individuals in the green, some criminal history, some without criminal history. They are not known to associate with each other, but they all take orders from this person. The Godfather. Do you think the Godfather was there when the crime was committed? Oh, heck no. Hey, I was at the movies. I don't know what you're saying. I don't know. I was there. That's terrible, Brando. But yeah, he's got an alibi, right? It's like I said, P53, VEGF. Like they can, a lot of times with cancers, if you think about it, they are affected by growth factors, right? Do the cells produce them themselves? Does the tumor? Sometimes. But a lot of times what happens is there's growth factors being produced other places that actually are affecting your tissue. And there's no way to measure the expression of those actually being made because you're actually looking at the tumor and not paying attention to the bloodstream going to it. Does everybody understand that? Something to think about. that there's a lot of factors involved in the cancer that you're studying that you will never measure because they're being produced other places. But with this technology, we can find them. And that's what I do. So this is just an example. Here's interleukin 1A. It's a cytokine. I never see it changed in gene expression. But in the databases that I use, again, we have criminal history on all these genes, right? What I can do is I can go in and say, okay, I may not measure interleukin 1A directly, but all of these things are previously known to interact with interleukin 1A, right? We get 43 targets of interleukin 1A in my gene list, right? The odds of that happening are 2 times 10 to the negative 17th. That's not random, right? Not only do I now know that interleukin 1A is playing a role in my system, but everything with an orange line, interleukin 1A is known to induce. Everything in red is going up in expression. Everything in green is going down. So all of these in the orange are known to be induced, and they're all going up. These two genes, interleukin 1A is known to inhibit, they're going down. The only genes that aren't following this upregulated interleukin 1A are these 4, right? And this is like 5.5 standard deviations away from what we would think is random. Basically, not only do I know interleukin 1A is there, but I also know if it's more active or it's inhibited, right? In this case, it's more active. Think of it this way. If I'm standing by a pond and I take a rock and I throw it over my shoulder, right? I might not know where the rock hit, but if I turn around really quickly and if I look in the water, I can see the ripples it made, right? And I can go find that rock based on the ripples it's making in the transcriptome. Genes do this all the time. And we can do this looking at microRNAs, right? We know a particular microRNA actually affects certain mRNA. We can look to see, we might not measure the microRNA, but if we see a bunch of things the microRNA is known to inhibit going down, we could assume that that microRNA is there. I'm finding something I'm not measuring directly, but I know it's there based on what it's doing to the genes that I know are going on. You can also do this with chemicals. So I always tell this story, and you'll see it in a lot of the videos if you ever watch my videos. I did this clinical trial for these doctors, this doctor group, and I'm just going to say right now, medical doctors do a great job with patients, but sometimes they can be terrible at designing their own clinical trials, right? They don't think of things. So in this one, basically in this, they were studying this pulmonary hypertension drug. And so they had 2 groups, and they were old men. In one group, they could only be on the hypertensive drug. And then they had a control group of elderly men that could be on anything except the trial drug. And then they compared the 2. So I went in and I was trying to find, an expression profile for the drug, and I didn't really see anything. But what I did see was something in the control group. So I saw a change based on a drug in the control group that wasn't in the experimental group. Does anybody know what that is? Elderly men, any guesses? What would be a drug that elderly men would take? Yes, vasodilators. By the way, that was awesome. You're dozing off and you're Viagra. Yes. So, we can totally, we know what genes are affected by Viagra. We've done plenty of studies. And in this study, when I'm looking at the data, I'm seeing all these genes affected in my control group by Viagra, which is not in the experimental group because they can't take their Viagra, right? Everything leaves a mark in the transcriptome. Everything you do. I can do drugs. It used to be, not anymore, but it used to be I could always kind of get an idea of a person's age or age group differences based on the amount of cannabis signature I saw in one group and not the other. And it was usually a young group. I would say nowadays it might be actually more in the older group than the younger group. Who knows? But yes, Everything you do, everything you take in your body, even the sunlight itself causes changes in gene expression that we can measure, right? And this is what I am using to basically try to understand my system. And again, we can do that with the VEGF that we found, right? We don't measure it directly. I do the criminal history. Angiogenesis totally fits in with tumor development, and it's been involved in lots of lung cancers. Again, I can look at my data. I find 51 targets of VEGF, which is definitely not normal. And not only that, but all of this information is telling me that VEGF is going up, which makes sense in a cancer. I can then basically take out all these genes and basically plot them over histology. So going from 0 to 7, so this is cancer, so this is the cancer getting worse. And I know this looks like a mess now, but then I can normalize it, basically divide everything by its mean. And now I get this curve, right? I'm willing to bet that that's the activity of VEGF. And I got that based on everything it interacts with, right? I'm not looking at VEGF directly. I'm seeing all the crimes it's guilty of stimulating and then using that to basically keep track of it. And again, we might be able to use this as a biomarker in that if your VEGF levels are high, that's not a good sign. You're probably going to get cancer. And we could probably use differences in concentrations to make clinical diagnostics, right? Right here, right? If you at stage 4 histology of your lesion, if you have high VEGF levels, not good. It's probably gonna go on to develop cancer. But if it's lower, probably okay. And again, we can find the genes in it. We can look at its crimes. Obviously, if you've got higher VEGF, you have a more chance to die than if you do not. So that's what we're really doing here. I feel like an investigator a lot of times. You know, you've seen those cop movies where they have all the criminals on the wall and they're putting the diagrams. I feel that's exactly what I do with genes, right? I'm putting these down. I'm like, all right, who's the kappa? You know, who's my godfather? Who are the wise guys? And I'm trying to put all that information together to form a story that I can actually use to inform my data or inform my study. So today what we're going to do, we're going to take a break first. I'm not going to make you go 3 hours all at once. We're going to look at triple negative breast cancer. And in the class that I taught before this, I was looking at, I think I was doing a form of brain cancer, but there was actually a student in the previous workshop that was working on this, and she basically brought up a gene that I thought was fascinating. So as we were doing our workshop, I was finding all this cool stuff, and so I'm like, all right, for this next workshop, we're going to look at triple negative breast cancer. So just to give you an idea what that is, triple negative breast cancers, it's not positive for estrogen receptors, progesterone receptors, or the HER2 protein. It accounts for about 10 to 20% of all breast cancer cases. And why do we care about this? Because it's more aggressive, right? If this was a form of cancer that was easily cured, probably wouldn't care much about it, but it isn't. And not only that, but it tends to affect minorities, people that actually probably don't get as good of medical care as other groups. So I think it's an important cancer, and so what we're going to do in our example is we're going to kind of study this, and I'm going to take you through some of these platforms and websites that'll help you not only generate data, but then help you understand what you're generating. This is kind of how bioinformatics works. As you get in, it's never, the farther you get into something, the more truth you get. It's just, you can go deep, 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 or you can go very wide. There's a lot of aspects to understand in this, and just realize that there is almost never a simple answer. There's always a complex thing, and that the farther you get into whatever you're researching, realize it's never going to be what the books tell you. It's always going to be a little different. And what I'm going to try to do today is teach you some skills to help you basically find that.